I'm going to talk about the biology, but also will describe the methods that we use to infer, to resolve the biological processes. So first question is, what are developmental processes? So developmental processes are the series of changes that cells undergo to form specialized cells. We can classify developmental processes to simultaneous processes, like in the case of blood formation, where when we do the snapshot of the tissue, for example, bone marrow, we can see all cell types. And also sequential processes, where when we do a snapshot at a given time, we will see only particular cell types. I will focus today about reprogramming. This is the process where we can transfer mature cell type to pluripotent cell. So we will reverse the natural developmental process and generate induced pluripotent stem cells that can be transformed to any cell types in the human body. So how should we think about those pathways, trajectories, landscapes? So the first person who thought about the process as a developmental landscape was Conrad Woodington with his metaphor of the developmental, or he called it epigenetic landscape. So he started with some constrained paths and described this as a deterministic process. But later he changed his mind and started to visualize this developmental pathways as a probabilistic process. And later he used this metaphor of the landscape, where at the top of the hill there is an egg or a marble that can roll down to the system of bifurcating valleys, where at the bottom are differentiated cells. And in the strategy of genes, he presented a vision from the behind of the cell, where the shape of the landscape is determined by the tension of interconnected guide ropes, which represent gene products that are attached to the pegs fixed to the ground, where the pegs are genes. So today we have an amazing tool that is called single-cell RNA-seq, and we can measure measure gene products, the RNA molecules in individual cells using next-generation sequencing. So the basic data structure we explore is a gene expression matrix, where rows are genes and cells are columns. And the element of the expression matrix tells us about the number of mRNA molecules in a given cell. And then using computational methods, we can classify cells to different groups. We can get the genes that are co-expressed in a given cell type. But the question is, can we infer developmental landscape using this technology? The problem is that single-cell RNA-seq is a destructive assay. It means that we need to lyse a cell, kill a cell, to measure RNA molecules in this cell. So we decided to test the reprogramming of fibroblasts to induce pluripotent stem cells. And we sampled this process at equidistant time points and measure RNA profiles. So we start from fibroblasts that we extracted from mouse embryos. And of course, the questions are, what are the cell types that emerge during the process? What are the trajectories? Is there a communication between different valleys? So we decided to generate high-resolution map of the process. Namely, we extracted the fibroblasts from mouse embryos using antibiotic doxycycline. We forced expression of four transcription factors, and we sampled this process every 12 hours along 8 days. Then we sampled every 6 hours, and then again every 12 hours until day 18. We also sequenced the mature pluripotent stem cells. And we published the results in the Cell this year. So the total number of cells we analyzed was 260,000. We had 39 time points, and the number of genes that we detected were 19,000. So first question is how to visualize the landscape. So with 3 genes, the visualization is not very difficult. But actually now the cell is a point in a 20,000 dimensional space. So we have, at that time when we analyzed the data, we had good methods for the visualization, but only for mature cell types. So in this strategy, first we do the dimensionality reduction because the data is very noisy. We capture only a few % of RNA molecules in a given cell. So in the first step, we apply the dimensionality reduction method in which we compute the principal components, and then we embed it in using TSNE to 2 dimensions. So as I said, this works very well in the case when we have fully differentiated cells. The real challenge is when we have a continuous cell fate and we want to infer the landscape. So again, we have like continuous changes in a 20,000 dimensional space. So what we propose is a different strategy. In the first step, we use nonlinear dimensionality reduction method that is called diffusion maps. And then each diffusion component describes given developmental process. And if we have a lot of developmental processes, a lot of cell types, we want to separate the diffusion components, and we do this by building k-nearest neighbor graph in the diffusion component space, and then apply forces, attractive forces to nodes that are connected, and repulsive forces to nodes that are disconnected. And we call this method force-directed layout embedding. And when we applied it to our data set, what we got is something that looks like a chicken. So we annotated cells by the phase. So in the first phase, we overexpress those factors. And then we change it to 2 different media, to I and serum. So the process starts here. Then at day 4, we see the broadening of cell fates. And then we have red is 2I phase, and blue is serum phase. And we visualize here 250,000 cells. So we can see that we have 3 major clusters of cells, red cells, so in 2I condition, and at least 1, 2, 3, 4, 5 clusters of cells in the serum at day 18. So what is very important, we can reconstruct the correct ordering of cells in time without knowing the time. So this method of visualization can reconstruct this very well. Of course, now we can use gene signatures. So gene signature is a group of genes that we know that are expressed in a given cell type. So for example, we can identify that induced pluripotent stem cells are in the chicken head. We have some trophoblast cells, neuronal-like cells, epithelial, and stromal cells. And we can plot the expression of those genes on this landscape. So for example, we see that on the West Coast, we see high proliferation. So that means that the cells that are there very divide very quickly. We can also know the ordering of cell emergence along the time. So for example, in the 2I conditions, we see that first population of cells that emerges are stromal cells, and then we have some IPCs, and they dominate the population. And in the serum conditions, we have most of the population consists of stromal cells, most of the cells consist of the stromal cells, and we have only small fractions of IPCs, because this process is very inefficient in the serum condition. But what we don't know the coupling, so we don't know what is the trajectory. So we want to find the trajectory that cells follow to IPCs. So, of course, this can be the trajectory like here, or this way, or this way. So we need to find a method of coupling cells from consecutive time points. So what we want to do is we want to couple any given set of cells to its descendant set at the later time points or to its ancestor set at earlier time points. So what we can use, and we want to model this process in a highly dimensional space of gene expression. So what we can use is we can apply the mathematical technique that is called optimal transport. Optimal transport was developed by Gaspard Monge in 1781, and the original formulation was how to optimal transporting a piles of sands from one place to another. It was later used by Napoleon in his campaign in Egypt in 1790s for building fortification with minimal work. So there is a difference between sand and cells. Namely, cells can divide and they can also die. So we incorporated the growth rates, and we use optimal transport, unbalanced optimal transport that was proposed by Tizard, where we use the entropy regularization. So we developed this package that is called Waddington OT. When we can compute optimal transport maps, we can compute ancestors, descendants, and also 2D embeddings to visualize the landscape. So now if we want to find the ancestors of IPCs, what we do is we just rewrite the table. So we assign the equal probabilities to all cells at day 18, and we follow them back, computing the transport maps. So what we can see that the successful trajectories on the west coast, and it goes through this tight bottleneck from this horn. So yeah, this slide exactly shows that the IPS population comes from this small fraction of cells at this horn of transformation. So because we know the probabilities for each cells to become IPCs, and we know the gene expression profiles for 19,000 genes, we can ask what are genes that are co-expressed. So here are genes. Red means that the gene is up in a given time point, and blue means that the gene is down-regulated. So then we can classify genes to different groups of genes. So for example, we see that at earlier time points, all the genes that are related with the cell divisions are on. And we can ask also other questions. For example, if there are proteins, transcription factors that regulate the expression of other genes that can be responsible for the process of this transformation. So we found one of the genes that is called OBOX6, which is an oocyte-specific gene. And what was interesting, it was expressed in a small proportion of cells only before day 12, but all of them were biased towards IPCs. So we decided to test it. We added it to the factors and express it, and we forced the expression of this gene along 80 days. What we found that indeed this gene can enhance the reprogramming efficiency by twofold in 2i conditions and sixfold in serum condition. On the East Coast, we have those cells that are called stromal cells. So we see the population of stromal cells here. Those cells are dying cells because they have apoptosis on, and they also show a very interesting phenotype. It's called senescence-associated secretory phenotype. It means that those cells produce a lot of cytokines, growth factors. They give the signaling, they give the signals to other cells to differentiate or to. Those cells, for example, are very important in the wound healing. So, of course, the question was, are those cells communicating with other cells in the valleys? So we tested this. So first, we defined the high potential interaction as if we see the expression of ligands in one cell types and the repressors that can read the signal in other cell types. So we asked the question, if there is expression of ancestors of stromal if there is expression of ligands in ancestors of stromal cells and contemporaneous expression of receptors for ancestors of IPCs. And when we did this analysis, we found that the top pair, it's GDF9, which is growth differentiation factor 9, and the receptor is expressed in the IPCs, and also on the string that we know that those cells transform, that are on the pathway to IPCs. GDF9 is slowly expressed, so this may suggest that this is somehow limiting step in the reprogramming. So of course we decided to test it. At day 8, we added GDF9. And what we saw is that it can enhance the reprogramming so here is the number of the colonies that we counted by eye, the IPCs colonies, and we see that this is dosage dependent. So the more we add GDF9, the more IPCs we see. And we confirm it also by other independent experiments when we measure the IPC signature. What we found is, so first we reconstructed the landscape. We proposed different methods for visualization and inference of cellular trajectories from time course snapshot of single-cell RNA-seq data. So we annotated all the cell types that emerged during the process. We found a transcription factor that can enhance the reprogramming efficiency. And we also found that there is a ligand, GDF9, that can enhance the reprogramming. And we simply added to the media, to the growing media, and we can enhance the reprogramming. What is interesting, there are 15 genes in this family, so from GDF1 to GDF15, and we tested all of them, and only this one works. So our predictions were very good, and we were able to find the correct one. So the next part of my talk, it's about the package that I developed. It's called SCSVA for single-cell scalable visualization analytics. So the number of cells we sequence today is very big. So we are very close to the big data. And this plot just shows that in 2012, there were only 18 cells sequenced at the Clark-Manser Observatory in the RegEF lab, but last year it was 19 million. So this matrices become very big because it's 20,000 by 19 million. And there is a huge initiative called Human Cell Atlas that now about 1,500 scientists are involved in the Human Cell Atlas project. And the aim of the project is to sequence about 1 billion cells across all human tissues to create the atlas of all cell types that are present in the human body. So what I propose is that we can use algorithms that we use to visualize from the astronomy to visualize the data from Gaia mission. And it's a very nice algorithm because we can explore very easily the embeddings and the cells. And this is in the real time on my laptop. So here we visualize 100 million cells. The only difference is that these data sets are not so highly dimensional as genomic data sets because we measure about 15 features, but here we measure about 20,000 or 30,000. So I added to the algorithm the fast methods of extracting rows of the matrices to visualize the cells even on a laptop. We also extended the algorithms in which we can visualize, we can build 3D FLE, and this we also added fast diffusion maps and also improved the algorithms for the force-directed light generation. So the cost, so all the steps on Google Cloud and the 64-core machine, it's about takes about half an hour to generate 1.3 million, the visualization of 1.3 million cells from the gene expression matrix at the cost of $1. And I would like to thank you to my supervisors, Aviv Regev and Eric Lander, and also to my colleagues that we work together on the reprogramming, Jeff Shibinger, Jian Shu, Brian Cleary, and Vidya Subramanian, and also to a lot of other people that were involved in the project. And SCSVA, I developed with Joshua Gold, and I would like to thank you, Anna Horsk and Lassie Gatney, for help with the graphics. And we are on the GitHub, so here our packages are available on the GitHub, and also on SCSVA is available in the Docker Hub. And there's also opportunity because I launched Laboratory of Single-Cell Genomics in November, and we'll have the opportunity both to do single-cell RNA-seq, measure the open chromatin, and also have the information about RNA-seq and information about the special positions of the cells in the tissue. And currently, I'm looking for 2 PhD students and software engineer to help me with the scalable analytics in the cloud. So we'll be developing methods for the scalable analytics, and we also will be developing algorithms for single-cell data integration, where we have the information not only about the RNA, but also about the protein, open chromatin. So you want to learn gene regulatory networks and also learn how to do the lineage tracing in the cells. So if you are interested, just shoot me an email. And thank you for your attention.
Thanks. Are there any questions?
Hi. I was just wondering, there's some fairly significant differences between reprogramming and just generally the continual growth of embryonic stem cells or iPSCs in humans compared to mice. So I was wondering if you think that what you've seen is reflective of what you might see in humans, whether there's a GDF9 homologue.
Oh, we didn't test it in the human cells, so I don't know. We don't have such a high resolution map of the reprogramming of human cells.